Yeah. Yeah. It's like, kind of previously the order. Cool. Okay. Cool. Go ahead. Hello, my name is Gabriel Siguenza, and then I'm a software engineering major and also part of the CTREE cohort for URES. Over the summer, I worked at, I did an REU with UC San Diego and SOC Institute. If you guys haven't been there, it's like really, really cool. It's across the street from the campus, and it looks like this. This is a picture of the actual SOC Institute, and it's a really cool laboratory. But anyways, I worked in the integrated biology lab with Dr. Graham McVicker, and we studied we were studying genetic variants on allele-specific chromatin accessibility. So again, this is kind of like outside of my field, so bear with me. I'm gonna try my best to explain it. But here we have like just a simple example right here of a SNP of DNA. You can see the like 4 bases. We have a single variant right here. We call it SNP, but right here, I think single nucleotide variant. I think we agreed that that was like a better term for it. So that's just one difference in a single DNA position. And we believe that these variants are might be causal to like diseases like diabetes, type 1, and other among schizophrenia and like other things like that. So so like I said, they're associated with human traits found in open. What I'm studying was that they were found in open chromatin regions of DNA. This is kind of like a new concept. So I'm going to explain that a little bit right now. There's a lot of moving parts. And it doesn't look like it right now, but all of this is actually in an effort to like work with a deep learning model that involves artificial intelligence, and like I'll get to that a little bit. But my part was just like part of measuring it. So there's a picture of our DNA. The double helix structure is very famous, but like we can see that when we rip them apart, there's these like previously thought fibers of chromatin that kind of make up the structure of our DNA, and it's also responsible for how tightly our DNA is packed together and also can be loosely packed. And these areas where it's loosely packed in this picture right here is the open chromatin, and that's kind of what we were looking at because we believe that, like it's believed that genetic variation occurs in open chromatin. When they're packed tightly, they're kind of locked away, and these are like kind of set, but when it's open, we have an opportunity for variation to occur. And there's multiple ways to like measure DNA. So like we're analyzing DNA. We have DNase, MNase, and ATAC. What I was studying was ATAC-seq data, specifically just that. And right here, I have it in a bar graph. These show the peaks. Those peaks correspond to where it's open in this DNA double helix where the chromatin is kind of loosely packed. And also, so by the way, this is a bioinformatics laboratory, and for those of you that don't know, that's an interdisciplinary field that develops methods and software for like basically analyzing biological data. So this is just a image that represents ATAC-seq. Here we have the closed chromatin that's tightly packed, the open chromatin right here, and we use this hyperactive TN5 transposase. I mean, I don't, but people in the lab do. And that binds to the open chromatin regions of the DNA that's kind of amplified in PCR sequencing to be able to see those fragments, and that's what you get those bar graphs from. So cool. And then yeah, so basically we can use this assay, which is just a method, to study the regions of accessible chromatin, and we're interested in studying the effects of genetic variants in these open chromatin regions, because this leads to like, we call it allele-specific expression, and that's when the genes are expressed that typically you end up with the end result, which is like, you know, disease or other things. So it gets complicated here because then I have to kind of explain this concept of allele-specific open chromatin. And I'm kind of looking at the DNA before it's like expressing these changes, but however, we have to kind of bear in mind that we get, we're diploid organisms, so we get a pair from a parental, like a maternal and a paternal. And sometimes we end up with this thing called allelic imbalance, and we're studying this allele-specific open chromatin with ataxi. And we're just recently coming up with methods that allow us to like measure these more closely. So just the recap is obviously I've talked about chromatin accessibility and ataxi. So when the expression levels aren't one-to-one, we call it allelic imbalance. And these can lead to issues, which I'm going to get into. So basically, when you do bioinformatics, you're mapping the entire human genome. And very famously, there's the reference genome that actually gets updated occasionally, and that ended up causing problems for my lab a little bit. So, so we have the reference genome, and when we use the reference genome to map our DNA to grab our data. And sometimes, well, all the time, it doesn't map correctly, and that's what we call mapping bias. And so we end up with that in our data a lot, and that's kind of what we were addressing in my specific portion of the project. And so that leads me to talk about WASP, which literally stands for WASP, allele-specific pipeline. And this is a pipeline developed by my PI. It was supposed to correct mapping bias and allele-specific open chromatin, does it very well. And this is kind of shows here, right, where we have a reference sequence and a non-reference sequence. We have a read, and we were not, like, when we map it, we're not really gonna be sure if it's gonna be an A or G. And so we're gonna take the best representation from the reference genome. So that creates like big problems because that's hugely misrepresentative of the actual patient's data. And so we had this idea that if we were looking at specific patient data and we introduced like a personalized genome, then we can get a more accurate representation. And we use this WASP pipeline to like narrow that down, and basically we end up with a much more accurate representation. And so this helps when we're studying things like genetic variants, because everyone has variations in their DNA. How would we know whether or not this is a person with diabetes or not, or, you know, we're gonna have issues because it doesn't map correctly. And yeah, so that's what I basically just said. It's highly biased. So this is the way that the software works. Basically, we map the read normally. There's tons of software to do that. And then we check if that read overlaps a single nucleotide polymorphism or a variant. And if it does, then we actually, it's kind of genius, we flip the allele and then we read it and we remap. And if it still maps to the same spot, then we know, then we know that we can keep it. But if not, then we'll discard the read, and it kind of goes through this whole process to do it again. And essentially we end up with that more clear read. So the data set that I actually used, which is really cool and you can only do if you have access to like a cluster computer, it consisted of macrophages derived from iPSC. They're basically stem cells for 23 individuals. And then we actually differentiated these macrophages. We like basically in the lab, they created these conditions that ended up with salmonella, which was one of them, and then I'm not really sure what the other ones are. And so that was the data that I worked with. We had to use like multiple patients to be able to train the model, the machine learning model, which I'll talk about right here. So in machine learning, I don't really want to get into it, but like basically right now we're looking at this new idea of deep learning. And deep learning is essentially when we feed an AI a ton of data, but we're not necessarily annotating everything and saying like, this is how you should interpret this is how you shouldn't. We let the AI kind of create its own relationships, and this gives us like a broader perspective of like maybe things we haven't seen or recognized before. And so like, this is just a basic example by NVIDIA. It's like a super famous company that created this convolutional neural network for genomics, where it feeds in basically phase haplotype data sequences, and then it goes through this convolutional layer, which you can read about if you're interested. And what we end up with is our output, our prediction. And so in the lab, a graduate student was developing their own machine learning model based on some, basically in the last like 2 or 3 years, we had discovered that deep learning is actually doing a very good job of predicting causal variants and in DNA sequencing. So we call, this is like a different study, like genome-wide association studies, but I can't really get into that with the time that I had. So basically, like we had these models that proved successful, and we were thinking if we introduce the personalized genomic data from our 23 patients, then we could get a further accurate, even more accurate, hopefully, or like be able to predict the effects. And so we hypothesized that if we, basically, like if we could attain higher performance, maybe not more accurate, maybe higher performance, but same thing, with these predictions by incorporating genotype and molecular function data from sequence variation that is present in the human genome, i.e. we know what caused this, we know that the patient has this affliction, so we feed in their data into the machine learning model. So our approach was to obtain measures of ASOC, which is allele-specific open chromatin, using that software. And then we wanted to obtain measures of ASOC, again, for the deep learning model that the grad student was developing. It's predictions, essentially. And then we wanted to compare the two in order to kind of gauge how well the model was performing. And so that's why I had to use the WASP pipeline, because otherwise we wouldn't know if the predictions were correct or not unless we actually map them using that pipeline. And a majority of my project was right here. I know it's just one square, but it was a lot. It was a lot of work. I had to map it initially using a software, right? And then we had to find the intersecting reads that have mapping bias according to the WASP software. Then we have to map it again, and then we have to filter out the duplicates. And the issue was that the software was written in 2015. There were not really any instructions, so you're running through it and I'm having these problems and we don't know what's going on. Nobody knows what's going on. Well, it turns out that they changed the human reference genome again and again, and so we're using an outdated reference genome, and so it's just missing large amounts of data. And so I ended up having to go through all 23 patients' genomes and convert them into the newer reference genome, and so that's what I did. So I did that, and then we fed it through the model again, no problem, worked out. So then we can move on to the next step, where actually the first WASP pipeline didn't have this step, which was the allelic balance count and analysis. It's also written in C, and it's a very older software, so it was had some just little things that aren't just intuitive to work with, and we have better tools now. So they had a WASP 2, however, when they rewrote WASP, they didn't rewrite the part that I worked on, which is why I had to do it. Then I was able to run it through the WASP 2, and eventually that will just be one pipeline. And so then at the end, we end up with our results, which would be the new mapped reads with no bias, right? Or presumably, there's some bias. And then I did all that work just to kind of get these little scatter plots so I can kind of show you guys. This one's just the scatter plot for just one of the conditions, salmonella. And you can see that on the bottom, these are the red, and the darker it is, shows that it's mapping to the reference allele, and then on the other side we have the alternate. It's a little bit easier to see in the alternate plot, because here we can see that over on the blue, these are significantly higher reads from the reference allele, and the red are the significantly higher reads from the alternative allele. And what would have been nice is if I could show you the same plots from before we ran it through WASP, because this is after. And so basically the grad student didn't finish developing the machine learning model because it turns out that that's pretty hard to do. So eventually we're going to run the ASOC counts and the WASP pipeline across the entire data set. I actually ended up being able to do that part, but then eventually we need to run it over the convolutional neural network that the grad student was developing so that we can compare the results. I still kind of am involved with SOC a little bit as an external collaborator, but for now, in the meantime, we don't really know how long that might take. It's going to be a long project, but it's really, really, really cool. And I just wanted to thank SOC, CTREE, UC San Diego, really cool, California State University, San Marcos, my home, and and Graham McVicker, my PI, he's a really cool guy, and all these people I met, and they were amazing. And over the summer, Fred Monroe is actually a consultant for machine learning and AI, and we actually met over Zoom, and he's in Canada, but it was he was there, he's family, it's really fun. And obviously the CSD STARS staff and URES. sorry, CTREE. Wait, no, you read. Dr. Garcia, Dr. Amanda, Dr. Ray, everyone, thank you guys. It's an amazing experience. And that's it. Thank you.
Hi.
Hi.
How's it going? So the whole point of the project is to try to create better like machine learning or like to detect the variance. Is that what
Yeah.
the point is, to then do some like more personalized like care for like patients?
Yeah, well, actually, we were interested in looking at the variance because we think that we can maybe isolate and find causes of certain things. So, like, we don't know, like, especially like with the large genomic landscape, like, we don't know what causes TB1, we don't know what causes like sickle cell and all these other like genetic diseases. So, when we started to realize that open chromatin was contributing to it, we found something that we could measure, and our tools are just getting better and more accurate. So I think eventually, like, it just seems like intuitive to start using machine learning or deep learning to reveal more relationships between these things. So I think ultimately we want to get a better performing model, and then I think we would start to look more carefully at like what caused it. I think the ultimate goal is to do it in silico. Like, a lot of things also that machine learning offers us is we might be able to synthesize data better and then end up not having to do it in the lab. But that's like way future. In the meantime, we'll do it in silico and then kind of reverse engineer it like in vivo and see if they correspond or not. Thank you. Yeah.
Oh, you wanna go?
No, go for it.
If it relates, you could go. That's it.
Sorry, I've done like very like basic forms of bioinformatics, and we used like an open access platform to basically run run things against a genome, like an annotated genome for us. So how is this a little bit different than what's already like accessible?
Well, I believe that the 23 pages are open. Like, I think we grabbed them from like the UK, like some database over there, and then we ran them through there. I think it might be different depending on where you did it. It just depends on like computational power also, because some of these things would typically take, I think, like 3 weeks to run on a normal computer, and we're able to like run them in 3 hours depending or whatever. It just depends.
Yeah, that makes sense, because like running stuff that's like just for a small insect actually took like hours and hours.
Yeah.
You're in like a human trying to annotate that.
Right, and so like, and also so like I've looked into it a little bit. They have this biophotonics core and like also this thing that supports their high throughput sequencing at SOC. So like they have like a robust cluster computing system, and so that like allows us to kind of utilize multiple cores to be able to run this process a little bit faster than typically you would be able to somewhere else. But not only that, but they also have this like GPU cluster, and that's going to be for the machine learning part, because those ones, we ran it for like days and then just like had it maybe kind of stop in the middle, and we don't know, like we never were able to interpret the results, so we started increasing our GPU utilization. So that's kind of like what we're looking at right now.
That's really cool.
Yeah. And you had a question?
Yeah, so you talked about how NVIDIA generated this pipeline for like the introduction of the convoluted data to yield this prediction, right? And then you start talking about how deep learning is gonna start like, or allowing for machines to make relationships with data by itself, like allowing it to make connections and those things by itself. I'm just curious how would that work if the machine has no like fundamental understanding for like what it's seeing, right? If there's no parameters for like, hey, this is what this is, and then also how do you utilize that in a research context if maybe the relationships that it's making inside of the the mechanism aren't the same as the person using it, right? So the interpretation might be different. Like I'm just kind of curious, like how I don't know computers. I don't even know how to.
Right.
work my own
Yeah.
stuff.
Well, no, it's actually a really interesting question. I think it's like outside of the scope maybe, but one thing that's interesting. I mean, like you could look into these things called generative adversarial networks, which is something I did a previous presentation on, but they're like basically machine learning algorithms that train each other. And like, I don't, it's just like a crazy, you know, thing, but yeah, but what was I gonna say? I forgot now. Yeah, so like, I guess that veers into this territory where we're trying to like create artificial intelligence that isn't more like, humans are biased, right? Not the computer. So like, humans create bias in that data, and the computer might reveal relationships that you don't even want to acknowledge. So like, that's kind of the reason why less human interaction makes the computer a little bit more accurate. But also like, we're looking into this territory that's like called causal inference. And that's a little something different because that's asking the question like, how do we learn? Why do we understand things? If I see a cat walk across the room and go behind a table, I can assume the cat went behind the table, but a computer can't. But now we're starting to figure out ways to like get computers to understand that, and that's like new stuff.
Yeah.
So
Okay.
I don't know.
can talk about this. You go ahead. You can answer.
Yeah, sorry, I'm not like the expert, and I don't know if that answered your question, but that's just
No,
my
yeah,
input
that's
on it.
Thank you.
Anybody else?
I don't have a question really fully formatted, but so the reason why to use the computers to so human bias won't be in there, but isn't there like I can't pronounce it.
Algorithmic bias?
So what about that? Are you guys taking into account, especially if you said that there's no like parameters, like how is it being defined? Like how are you getting data? How do you know that there's no algorithmic bias?
Yeah, that's a good point, actually. Yeah.
It's not being defined.
Yeah, I think I might have misspoke. We weren't necessarily using that methodology, but like we're using something where it's like, instead of like we remove the characterization portion, that's deep learning. Deep learning kind of starts to create the, like you do have to give it some sort of information, and then the computer starts to like annotate it based on that. But like going further than that, and that would be something else, and like then we would introduce algorithmic bias. But in our case, I think we were just utilizing this like new form of machine learning. Like deep learning is just like basically machine learning 2, like version 2. It's like better. So yeah. But I mean, that's a good question though, and I don't know the answer. Like there might be bias there too as well in the algorithms, which is why they're still developing the pipeline. And WASP has been cited in a lot of papers like now that are using it as a tool to like achieve their goals and improve their software. So it's just a starting point, I think. Anyone else? No? That was fun. Oh, you have one?
One more question?
Yeah.
I just want to jump off that one. It's a great question. And so it has me thinking then, so you use the reference genome. I think the reason for that is just the amount of coverage that has, right? This is probably one of the most like best sequence versions of the human genome that we have.
Yeah.
Well, now that we have like 1,000 genomes and like other ones that are starting to approach that same amount of coverage, would there be any like advantage, disadvantage, like no difference in using some other genome as a reference? Should we consider like other populations that can be used?
Yeah, no, I think that's a great point and question. I think that there should be, and eventually that we would want to consider a lot of those different like populations, because I don't know who the reference genome comes from, but I mean, I'm sure he's not representative of everyone. And I think like the way that they are designing the pipeline, the way that they're working on this, like their deep learning models based on like this Genomics Informer and Basenji model, those ones I don't know that this is true, but like you should be able to kind of interchange those parts and kind of still get a better idea. Then you can compare the results across all of them. I think that would be the goal ultimately. But we needed a solution in the meantime because we only have so much like that we can use as a reference. But we still need the reference. Like yeah.
No, because I think about like even trying to incorporate all the like the common SNPs that exist to account for any sort of biases that would just come from that reference. But yeah, you say AI, like I just wanted to recognize.
I agree. Yeah, I think it's interesting that before we were reading data using the human reference genome, we were mapping it, and like there was so much information missing. So like it's crazy that I mean maybe people didn't even know, so they drew conclusions on data that was like incorrect. And so that removing the mapping bias was, I think, the first step into getting a more accurate interpretation of that data, which is why bioinformatics is such an interesting field. Yeah. Any informatics. Yeah. Cool. Can I leave now?
Cool. So 3 more great presentations. Hopefully you all learned a lot.